Forget frequently asked questions.
Common sense, common knowledge, or Google.
How about advice from a real genius? 95% of people in any profession are good enough to be qualified and licensed. 5%
go above and beyond.
They become very good at what they do, but only
0.1%
are real geniuses. Richard Jacobs has made it his life's mission to find them for you. He hunts down and interviews geniuses in every field: sleep science, cancer, stem cells, ketogenic diets, and more. Here come the geniuses. This is the Finding Genius Podcast
with Richard Jacobs.
Hello, this is Richard Jacobs with the Finding Genius Podcast. I have Shinyi Marzano. She's a molecular biologist at USDA. And we're going to talk about RNA silencing pathways and her work. So, Shinyi, thanks for coming.
Thank you.
Yeah, please tell me about I know you recently moved to the USDA. Tell me, what's your work there about?
Yeah, I work under ARS, Agriculture Research Service, which is the powerhouse for USDA research. So I focus on developing tools for integrated disease management programs. I develop non-chemical disease management and other pathogen detection approaches. So we use genetics, genomics, and molecular biological approaches. And we focus on greenhouse production or high tunnel production, which are both controlled environment production.
What kind of plants do you focus on?
So I should rephrase the question here. I actually focus on pathogen biology, and pathogens we focus on tend to attack dicot plants. So crop plants can include lettuce, tomatoes, strawberries, and beans or peas, all the dicots. So we work on understanding the pathogen biology and the mechanisms of plant resistance.
And what is a monocot versus a dicot again?
Yeah, dicots are the ones that produce dicotyledon. So, like say, if you grow your beans from home, wet paper towels, then you will see two leaves coming out, and they are the dicots versus rice or other grass species or corn that they are monocots.
Also, the first green shoot that comes out of the seed, it'll be either one or two shoots. So the ones that come out with two are dicots. The ones that come out with one are monocots.
Yeah, they have two cotyledons. So the pathogens we work on mainly attack those plants. And so white mold or gray mold, they focus on attacking those crop plants. And they cause a great loss in yields annually.
Okay. So yeah, these two molds, the white and the gray mold, I guess these are fungi. And where do they come from? Like, where do the spores come from to make the mold if you're in a greenhouse or a controlled environment?
Right. They can be brought in through, water, because even controlled environments, oftentimes we don't use purified water, but tap water, and also air can bring in the spores. So since they are very persistent in nature, so they can just come through to the greenhouse and other controlled environments.
So what happens when a plant gets white or gray mold? Do you see it pretty quickly, and what happens to the plant?
The plants generally are very susceptible during their flowering time, and so the spores, mainly the sexual spores, ascospores, will germinate when they are attached to the flowers, petals maybe. And so then they form mycelia throughout the plants and become a systemic infection. So they can spread throughout the plants and then down to, so causing a lot of times stem rot and for white mold, basically. But for gray mold, botrytis, they can be dormant on fruits. And then, so when you go to do grocery shopping, sometimes you don't see them moldy, but after you bring the tomatoes back, or strawberries mainly, then after a few days, you see some gray fuzzy growth. And that's gray mold botrytis. And so they can just, humidity increases the chance of their infection. So like when they are even in the fridge for a period of time, just a few days perhaps, then the spores can germinate and colonize the whole fruit.
I thought in a refrigerator it's usually dry, so it would make it harder for spores to do anything.
Yeah, maybe the packaging increases the humidity because like strawberries are often housed in plastic domes or in their packaging.
Yeah, that's true, right? If you leave stuff and it's in plastic, it does get moldy pretty quick.
Right. Yeah, so generally speaking, the most we are working on, they lack humidity. And so what we are trying to develop is using one that is a virus infecting these fungi, and second, RNA silencing approach to target essential fungal genes so that they are not able to attack plants anymore or delay the attack before you can consume the fruits.
Are fungi studied pretty heavily, or I don't think so? I don't hear much about them. Like, have these fungi been sequenced? And I have a lot of questions about it.
Right, yeah.
Is the sequence of mycelium different from the sequence of the fruiting bodies? Have you ever tried that?
Well, for genomic sequencing, the genomes are consistent, so it would be the same. And then both white mold and botrytis, I believe, too, are sequenced for their genomes. And then certainly, perhaps during different growth stages, they have different transcriptomes that are the genes, expressed. But for the genomes, they are sequenced and maybe not so well annotated. So yes, they are relatively understudied, and specifically, we are interested in small RNAs. And if you are aware of RNA silencing or RNA interference, which serve as both an antiviral mechanism as well as they play a lot of regulatory roles in most of the organisms. So small RNAs are not very well studied in fungi in general, and RNA silencing pathway, which is what produces the small RNAs, are also more understudied. So that's where we focus on trying to dissect the RNA silencing pathway of these fungal pathogens, because between plants and the pathogens, it's an arms race, and then the small RNAs are often serving as a virulence effector. And so when we determine what's important or the function of the small RNAs, we can try to disarm the fungal pathogen.
So yeah, how does interfering RNA or silencing RNA work? Can you give me a little bit of some info on it?
Sure. Yeah. So RNA silencing is triggered by double-stranded RNA, and so in fungi they are widely infected by a lot of different kinds of mycoviruses. We call they are fungal viruses. So viruses infecting fungi. And then so RNA silencing in fungi are triggered by virus produce the double-stranded RNA. So when fungi sense the double-stranded RNA, they will try to destroy the double-stranded RNA produced by the mycovirus. And then so the double-stranded RNAs are being chopped up into small pieces, and that's called the virus-derived small RNA. So that's a signature of RNA silencing in work as antiviral mechanism in fungi.
What's bad about double-stranded RNA? Does it occur in fungi naturally or only because of viruses?
So the majority of the double-stranded RNAs are from virus in fungi, and then as well as in other organisms. So our bodies also sense double-stranded RNA from virus infection. And so it's not part of the host, if you will, that would produce a large amount of double-stranded RNA. It's a signal of invasive nucleic acids.
Does double-stranded RNA have special abilities inside of a cell? I mean, it gets transcribed into proteins, because if it's double-stranded versus single, what special abilities does it tend to have?
Right. It's mainly a replicative intermediate for virus. So yes, it can further serve as a template for replication, but from the host in our context of study, it's the fungi, which is the host for the mycovirus. They sense it as invasive nucleic acids, and then they will try to destroy it. Yeah.
Okay.
So when we disarm the fungal host with its essential proteins that are part of the RNA silencing pathway, then the fungal pathogen will be very susceptible to any invasive nucleic acids, including mycoviruses. Apart from there, also we noticed that those essential genes, which are disers, argonauts, and RNA-dependent RNA polymerases. So when some of these are missing, then there's regulatory deficiency or defects in the fungal pathogen. So that's where we find their weak points that we can target.
How do you prime the plants so that they can more effectively silence the RNA from fungal attacks? How do you give it to them?
That's what we are actively working on, because recently there's finding that sometimes either plants producing, if we can engineer plants to produce double-stranded RNA targeting the fungal pathogen, or just applying external double-stranded RNA on plants, and then the plants are more resistant to fungal attack later on. So it can be either generated by the plant itself, or we externally apply double-stranded RNA on the plant as a protective.
Well, you just spray it on in a certain form, and it gets absorbed into the plant.
Yeah, it can be either absorbed, and I don't know if people have found out the efficiency of absorption, or it can just stay on the surface and wait for the fungal pathogens to attack the plants. So maybe the timing of a spray of such a double-stranded RNA pesticide is important, so that we time it so well that say during the flowering time when the ascospore is about to germinate, then we can suppress the virulence of the fungal pathogen by spraying.
Well, instead of just spraying, why not put it in the water source? Would that work?
Yeah, I think that can produce a lot of double-stranded RNA economically. I think that can be protective. I think that's a good thinking. Now the question is whether double-stranded RNA is very stable, and that is a question we are asking too. And from the general understanding and our lab's previous experience, double-stranded RNA is almost as stable as double-stranded DNA, unlike mRNA, which is single-stranded. So it may work to put it in water for the plants to absorb, but I think this brings up another question: how efficient that is for such absorption into the plants.
Can you make a virus-like particle and make a capsid, or an envelope or a membrane for the double-stranded RNA to allow it to be protected more?
Yeah, that's a very creative way of thinking. We do have viral vectors, and we rescue mycoviruses that could serve as a carrier for double-stranded RNA. Currently, we actually we'll try to use E. coli to produce double-stranded RNA, and then such a E. coli strain that's missing exonuclease 3 will not degrade double-stranded RNA and instead keep it intact, and that's how we perceive that to be more stable than already chopped up small RNAs.
If you like this podcast, please click the link in the description to subscribe and review us on iTunes.
Well, there's probably a lot of vectors. I mean, do you let in pollinators, like bumblebees for the tomatoes and stuff? And maybe the pollinators themselves could have, the double-stranded RNA on their legs, and when they pollinate, they pass it. Or
right.
All right, so that's one question. Could you do that?
One mycovirus we are actively working on. It's likely to be transmitted by insects. So it's a fungal nets that probably transmitting it. I say probably just because we are still in some early stage of this research. However, there's a very similar mycovirus that's being studied for about a decade now in China. That other lab, they found out that fungal nets transmit their virus that's very similar to our mycovirus. And so I think it's possible to combine the two technology using, say, mycovirus, viral particles transmitted also by insects to spread the double-stranded RNA, or just simply the mycovirus, because we are involved in both facets of the research, which are very connected, like you have observed, because we rescue these mycoviruses from the environment, and then we use them to probe, if you will, to discover which RNA silencing genes are important for those fungal pathogens. So yeah, if there's somehow we can stack these two facets of research together, it would be something we can explore down the road.
Okay. And I thought, I've grown some tomatoes and some other plants in like a grow tent, and at the store they had, some sort of mycorrhiza that you pour into the plant's water. I did like a flood and drain system. But when I poured in this, this beneficial fungus, or beneficial mycorrhiza, it really helped the plants grow tremendously. So, from what I understand, plants all have like a rhizosphere, near their roots. Wouldn't the funguses that hang out there be beneficial in protecting them against other fungal attack?
Yeah, obviously, you may have done your homework on this. There are recently some new studies, also from the lab I referred to in China, that their white mold infected by one particular mycovirus becomes a beneficial endophyte that can promote plant growth. And so we are still testing on our mycovirus that's a little similar to theirs, which is a single-stranded DNA virus that's relatively uncommon in the group of mycoviruses overall. So they found out that their fungal strain that was supposedly white mold and pathogenic, now after getting infected by this single-stranded DNA virus, it's promoting plant growth. And even more, the fungal strain now can infect wheat and barley, those monocots, and then promote growth and increase the yields. We're not replicating their research on those, but we also are trying to see, besides growth promotion, whether there is other mechanism, say, increasing plant resistance to later on infection. So we've done some research, and it's not published yet, but we can see that there is some systemic acquired resistance involved. So if we, say, prime the plants with such a fungal inoculum with the mycovirus, now not pathogenic anymore, not a pathogenic fungal strain anymore, if we prime the plants with that, then plants are more resistant to later on infection. And so then we did RNA-seq and found out the genes that are differentially expressed, and mostly in the pathway of systemic acquired resistance.
Yeah, at least it's nice that you have a lot of different vectors that you could bring, the materials in on. You need, again, through water, through spraying, through bees, maybe through ants. Just all kinds of ways. So, I have a good feeling that you'll solve this problem because there's so many angles of attack to it.
Yeah. So yeah, we are being optimistic too, but also it's very intriguing to see how mycovirus may not be a backseat driver. Instead, it can reprogram the fungal pathogen to become non-pathogenic and even beneficial under some condition. So to understand how we can manipulate this change or transformation is the basic knowledge that we can maybe apply across board to try to find a tipping point when these pathogens are not pathogens anymore.
Well, yeah, studying these white and gray molds, what kind of viruses prey on them? What kind of bacteria interact with them? I'm sure there's there's a ton of interaction that happens with them. And if you can influence one of their partners, like you said, viruses, to transmit some genetic material to them to turn them beneficial, or again, maybe bacteria that interact with them and will change them. Again, there's a lot of angles of attack there, you know?
That's true. A recent study also published just a couple months ago by the same group in China. They found that like you described, the microbiome surrounding those primed plants are different. So that they did some microbiome analysis and found that the functions of the microbiomes are more tightly connected. So which probably contributes to why there's increased growth in plants. So yes, microbiome, bacteria interacting with, mycovirus infected fungal pathogen, which now colonizes the plants in a harmonious way. Then they have a different microbial function overall.
Yeah, have you found any beneficial fungi that would keep out the white or the gray mold?
Right. Should say we currently focus on our system, which is the single-stranded DNA virus we discovered through metatranscriptome that makes white mold no longer pathogenic. So then we utilize it to help with plant disease resistance, like I described the systemic acquired resistance, mainly currently in soybean. And we want to see if that happens to other plants as well. So we have some strawberries planting in our greenhouse, and we'll do the same, and sunflowers too. So see if the same mechanism in plant resistance apply to different crops.
Okay. Well, very good. Shinyi, so I don't know, what's ahead for your research over the next year or two? What do you think you're going to figure out, or what are you close to figuring out?
We'll be doing more of greenhouse work studies, and previously we focused on more assays that's on leaves, but now we can focus on whole plant treatment. So if we show that the whole plant treatments are effective, both for our mycovirus biocontrol application as well as our RNA silencing approach, then we available tools to public, and that's generally what our research goals are in USDA ARS, that we want to be relevant to our needs, the needs of our stakeholder. So working under this current unit, which is called Applied Research Technology Unit, we try to develop tools that are applicable to growers and producers.
Okay, very good. Well, Shinyi, what's the best way for people to find out more about your work? Where can they go?
Usually ResearchGate or Google Scholar. That's where people can find our publications. And I'll try to post our progress on Twitter as well.
All right, well, very good. Shinyi, thank you for coming on the podcast. It's been very interesting.
Thank you for having me.
If you like this podcast, please click the link in the description to subscribe and review us on iTunes.
You've been listening to the Finding Genius podcast with Richard Jacobs. If you like what you hear, be sure to review and subscribe to the Finding Genius podcast on iTunes or wherever you listen to podcasts. And want to be smarter than everybody else? Become a premium member at findinggeniuspodcast.com. This podcast is for information only. No advice of any kind is being given. Any action you take or don't take as a result of listening is your sole responsibility. Consult professionals when advice is needed.